Hello, I'm Jean Greenberg, President of the International Society for Molecular Plant Microbe Interactions, or IS-MPMI. On June 17, 2026, IS-MPMI inaugurated a new webinar series called Road to Jeju. And our first speaker in the Legends of IS-MPMI portion of the series was Roger Innes. If you didn't catch the webinar, you can watch it on our YouTube channel or on our society website in a few weeks. The response to the webinar was overwhelmingly positive and generated a lot of great questions. In this session, we'll talk to Roger and ask him some of the questions we could not get through during the actual webinar. I'm joined today by board members Georgina Fabro and Minu Singla Rastogi, and we'll take turns asking Roger these questions. Oh, before I go to that, I'll just also want to acknowledge our sponsors who made this series possible. And you can learn more about them by going to their websites or using these QR codes.
Okay, let's start. We have grouped the questions, the remaining questions, into 6 thematic groups. So this is the first one, the first topic about surface RNAs and their potential use in agriculture. And to follow up on this, and Rao Upapalati's question about challenges of developing commercial products, Pawan Kumar asks, do you see a future where RNA can be utilized to develop RNA-based fungicides or other commercial agricultural products?
Yes, absolutely. In fact, there are multiple companies I'm aware of already that are trying to develop RNA-based products, especially fungicides and targeting insects. Those are kind of the two big targets that I'm aware of right now. The challenges are just the stability of RNA on the leaf surface, and the sort of because of that, the environmental resilience of that treatment and the cost of producing the RNA. But there are solutions to those challenges, especially new ways to package the RNA, ways to modify the RNA to stabilize it and have it more efficiently taken up by the target organisms. So I do think quite soon we'll be seeing commercialized products that are based on RNA.
Could I follow up that?
Sure.
Because that's all assuming like uptake of the RNA, but some of your results suggest that maybe external RNA could also have some effect. And is there any idea about using that?
So to clarify, what I was just talking about was external RNA. So the so-called spray-induced gene silencing is one example of that application. And what we're learning from the surface RNA that is produced naturally by the plant is I think that's going to give us insights into what sort of RNA structures are the most stable on the leaf surface. And we can use that information to stabilize RNA that we spray on the leaf surface. And also, I think another question that's going to come up here is what stabilizes the RNA on the leaf surface naturally? And we're starting to look into that. And I think once we understand that better, that will guide us in how to prepare the RNA that we actually spray on leaves.
Great. Okay. Minu is going to take the lead on the next group.
Yeah, thank you, Jane. So Rasher, the next topic that we want to touch upon is how to define the specific molecules that are responsible for immunity and distinguish the sample types. And looking closely at the vesicles themselves, so Sungil Kin asked if there are any specific markers that are available and can be confidentially used to distinguish the leaf surface wash from apoplastic wash fluid?
Yeah, we've done a little bit of work on that already and actually published some of that in our PNAS paper last summer. The proteins that we find that are abundant in apoplastic wash fluid, such as PR proteins, PR5 being a really good example, are abundant in apoplastic wash, but we see hardly any of it in the leaf surface wash. So I think you can distinguish leaf surfaces from the actual apoplast by proteins that are just secreted through a standard secretory pathway. And so right now it's a PR5 is our best marker, but I think you could use most any of those classic PR proteins for that purpose.
Roger, for the non-plant specialists who might want to look at your work a little more, could you explain what an apoplastic wash is? Like, what is the apoplastic? Just for people who may not be
Yeah, of
so.
course. So the apoplast is the term plant biologists use for the intercellular space in a leaf, especially. And a leaf is actually full of lots of airspace, and that's to promote gas exchange. But you can flood that airspace by submerging a leaf and applying a vacuum and then releasing it. And so you replace all that air with a buffer, and then you can recollect that buffer by a low speed spin that's kind of like squeezing a sponge. And so that fluid comes back out through the stomata, the natural pores on the leaf surface. And that buffer then that we recover from washing the intercellular space in a plant is what we call the apoplastic wash fluid. And we call it wash because you are flooding what normally is an airspace with buffer and then recollecting it. So you're kind of rinsing the outside of the cells that are inside a leaf.
So just to follow up on that, Sangal also wants to know what specific EV cargo, be it proteins, lipids, metabolites, or cell wall materials, are primarily responsible for delaying bacterial and fungal growth.
Yeah, I don't think we have a good answer to that question yet. I don't think anybody does. My lab has been mostly focused on RNA, and we know that RNA is mostly outside the EVs, so I wouldn't call it EV cargo. I would call it cargo of EVACs, so that the large 1 to 5 micron vesicles that we were describing at the second half of that talk. So there we know that at least for inhibiting growth of bacteria, RNA can contribute to that. But we also know that EVs and EVACs contain glucosinolates, working with Arabidopsis. Those glucosinolates are a secondary metabolite common to plants in the crucifer family. And those are known to have antimicrobial, especially antifungal activity. So it's probably a combination of metabolites and RNAs that contribute to inhibiting both bacterial and fungal growth. It's not just one or the other. But the experiments really haven't been done to see which contributes more, and it may vary from one microbe to another as far as what it's most sensitive to. Lots of work to be done there.
So EV is the extracellular vesicle, and the
Yeah.
EVAC are the larger.
That's how we're using that
Yeah,
terminology
okay.
currently.
Just
So
to make it clear, and if people go back to the seminar, they can see beautiful images of those different structures.
yeah, so there's many publications now on extracellular vesicles from plants, but these are all sort of under 200 nanometers in size. And our data so far with EVACs makes it look like at least some of those carry EVs inside of them. So this may be a way that EVs get out of the plant cell is inside these larger vesicles that we call EVACs.
Okay, great. So I'll leave this topic. And this is a group that's grouped into mechanisms of secretion, stability, and stress. And some of these questions come from myself and Georgina, and also participants in Chil, Gio, and Martaza Golem. And the topic is how plant stress alters RNA EV production and how secreted RNA remains stable. This is related to the other question. So this combined set of questions also includes the extraction process in addition to secretion. So first of all, how does secreted RNA maintain its stability on the leaf surface? And are components of the vesicles involved in stabilizing?
So let me answer that first one. So what stabilizes the RNA on the leaf surface? Our leading hypothesis is that the RNA forms condensates with polysaccharides on the leaf surface. And our evidence for that is that when we purify the RNA through a standard trizol purification method, we get lots of pectin and other polysaccharides co-pelleting with the RNA. And we also can show that if we use eGTA or eDTA in our buffer, which chelates divalent cations such as calcium and magnesium, we actually can extract a lot more RNA from the leaf surface. And so we think a lot of the RNA is kind of stuck on the leaf surface because it's associated with these condensates with the polysaccharides. And by chelating the divalent cations, you actually break up those condensates and release a lot of RNA. So we think that's probably the best explanation for the stability. We've got some work to prove that, but the data is certainly pointing towards that. So components outside EVs involved in stabilizing it. Well, I guess that's really talking about the polysaccharides and the divalent cations.
Can I make a follow-up on that?
Sure.
That since you mentioned pectin, so the pectin monomers will travel into the EVACs together with the RNA, or it's outside that the RNA will bind?
We assume it's only after the RNA is released from the EVAC, so it would be associating with the pectins.
Yeah, because pectin is secreted via Golgi, so it won't
Right.
be in the vacuole, like, okay.
Right. Yeah. So the general model is that these EVACs get released but are quickly broken, and then the contents so the EVs that are inside the EVACs and the RNA that's inside the EVACs then would spread out on the leaf surface, and that's when association with the polysaccharides would happen.
Great. So this question came from me. I wondered if maybe you said this and I don't remember, how much of a time course you tried for the leaf washes. And really this goes to whether you think there's truly a continuous deposition of RNA on the surface, or could it be like when you do a wash, it's just inefficient and you only remove a fraction of what's actually there?
Yeah, going into this work, and I should say this is Mina and Lucia that did these repeated wash experiment. We expected that we would be able to deplete the RNA on the leaf surface, and I think they did 3 or 4 rounds. And recognize it takes about 30 minutes to go through the whole process of rinsing it and recovering the RNA. But we didn't see a decrease in the RNA that we would get with each wash. And I would think if it was just inefficient, you would see a notable decrease from one round to the next. The one problem we had was after about 3 rounds, you start seeing some damage to the leaves. And at that point, you don't know if the RNA is coming from broken cells or just from the leaf surface. The other reason, though, I think it's probably continuous, was just how fast we saw an increase in RNA on the leaf surface after SA treatment. So we could get 2 or 3 times as much RNA from a leaf surface prep just 1 hour after SA treatment, which tells you that RNA is coming out pretty quickly.
Right. I think one of my questions didn't make it into here, but I'm going to ask it. I'll take my prerogative to ask it, which is whether you discussed the potential role of this RNA and the vesicles in affecting microbiome assembly. And I'm wondering if the opposite could also be true. Is because even though you're using like basil leaves, they're not sterile. So I wondered if you had tried some aseptic plants, and do you see like the same kind of RNA on septic?
Yeah, no, that's something we're really curious about, and we haven't had a chance to do that experiment yet. But we actually proposed that in a recent grant application that's pending review, was to grow what are called gnotobiotic plants, so plants growing in the absence of any microbes, and see if the RNA on the leaf surface differs from what we see on just a normally grown plant. My guess is that there'll be a big difference, but we haven't done the experiment yet, and we hope to do that soon.
Yay, okay. So let's see. I think we already did this. Okay, yeah. So building on your statement about salicylic acid spraying and the increase, do you think plant stress, wounding, or infection change the composition and sequence profile of these RNAs and EVs, suggesting a targeted secretion, or is it simply a general increase in constitutive secretion?
Yeah, I would say our data so far is most consistent with the latter, that it's probably just a general increase in constitutive secretion when you apply stress or spray with SA. Certainly, you can identify specific RNA sequences that go up relative to the general population, but I think that just reflects an increase in those RNAs in the cytosol. So, specific microRNAs are increased in response to stress, and then we would also see that as an increase on the cell surface. So, yeah, at least at this point, we don't have any strong evidence for real selective packaging of RNA in these evacs.
Let me ask you about specificity. You said that this response happens like within an hour.
Mhm.
Like, do you think any mild acid or like how specific is the response to SA particularly, or maybe other defense molecules or just, a change in the pH or whatever?
Yeah,
Um,
we haven't carefully looked at that. I mean, we've done JA and SA. I don't think we've done any other plant hormones. I don't think we've tried particularly well. I guess one thing we have tried is different pH buffers. And with a higher pH buffer, we get much higher RNA yields. But our interpretation of that is that the high pH buffers inhibit the endogenous RNases. And so that you're just getting overall increase in RNA yields. But I don't think we could rule out that the high pH is just a major stress inducer too.
yeah, I mean, one thing that's nice to do is to take an active compound and an inactive, like from the point of signaling, if you think it's a signaling effect. So you could, I don't know if there's some salicylic acid-like molecule that isn't
Mhm.
evoking the usual SA pathway, uh, or whether that would be a way to distinguish that.
Um, yeah, no, that's a good idea. We could also perhaps try a receptor mutant. So
Yeah,
something.
yeah, that's another way to get at the specificity.
Mhm.
Although the thing about receptor mutants is like they've never had a response, so they also have constitutive differences.
Yeah,
Oh, there's different. I think there are different benefits from using active, inactive, and also
Mhm. Yeah,
receptor.
that's a good suggestion. We
You
haven't
know,
thought about.
different ways of getting at the converging on the same thing. Oh,
Mhm.
okay. So I think we're back to menu now.
Yeah. So here, Rasha, we will be switching gears a little bit and talking about the interaction with the microbiome and the microbial community. You already touched upon those topic a little bit, and but here, some of the folks like Parik Habanga and Sarah Heller-Marian are curious about the communities on the leaf. So they want to know what was your reasoning behind building the synthetic community with those specific microbes that you mentioned in your talk?
Yeah, so that was just a combination of practicality and also wanting a diversity, phylogenetic diversity of microbes in the community. So the practical aspect of it was we had a limit to how many different microbial species we could culture. So I think this was a collaboration with Ken LeCorbin's group at University of Kentucky, but they streaked out and restreaked and colony purified about 100 different plates that represented colonies from the maize leaf. And then we sequenced those and identified, what they were. And then we compared that to our RNA sequencing data to see what was present in the total microbiome from the RNA sequencing data. And we found some interesting disconnects between what was present doing RNA-seq versus what was present doing DNA-seq. Because we also isolated total DNA from the leaf surface. And so part of what we selected in that synthetic community were microbes that were abundant based on DNA, but seemed to not be very metabolically active because they had very few reads in the RNA. And so they're sort of dormant on the leaf surface. And we thought that those might be ones that were more sensitive to RNA, and therefore their growth was being inhibited. And so that's in fact what we've been looking at so far. And we do see a correlation, but we need to do more microbes to see if that correlation is going to hold up. But it does seem like the ones that were metabolically less active were the ones that take up RNA better.
You have a follow-up question. Is it feasible to do fish on these leaf surfaces to see, compare the ratio of like DNA to RNA directly?
We've discussed that. We haven't attempted it yet. You know, with all the spatial transcriptomic tools that are coming out, and you know, if there's some way we could fix the bacteria onto the leaf surface, maybe we could do that. But I think technically it'll be challenging, and we haven't had the time to do that yet. But it's an interesting question.
Well, we can like dream about the experiment
Yeah,
we want to do.
yeah.
Your dream experiment may not be immediately feasible, but
But that would definitely be a dream experiment, would
yeah,
be
yeah.
to
It's good
essentially
to have those dream experiments.
a spatial transcriptomics of a leaf surface.
Yeah.
So just following up on that, so can leaf surface RNAs act as signals that influence which bacteria successfully colonize the phyllosphere under abiotic stress? So what's your thought on that?
I would expect that it would. I mean, again, we haven't done any abiotic stress experiments, so we have no data, but I would expect that abiotic stress, similar to biotic stress, would increase the amount of RNA on the leaf surface, and that we then would predict would change the bacterial community on the leaf surface.
Yeah.
And whether that resulting community makes the plant more stress tolerant or not, this would be a really interesting question to pursue.
Yeah. Indirectly not, directly not, but indirectly maybe we have because of the heat stress in the growth chamber sometimes.
Yeah, yeah, that's true. Abiotic stress there.
Unscheduled
As well.
experiments. Yeah.
Yeah, unscheduled. So, is there any noticeable RNA profile similarity between the leaf surface RNA and leaf isolated bacterial species? I think this is my most favorite question.
I have to say, I don't fully understand this question, whether it's asking whether there's a correlation between the RNA profile and the bacterial species found on the leaf, or are they asking about RNA in the bacteria?
Yeah, it is a mixed question, but I would like to interpret it as whether these leaf surface RNA have direct targets in the leaf isolated bacterial species.
Okay,
Or
so that would make sense. So yes, we think they probably do. And so what we've been doing along those lines is some bioinformatics to look for genes in bacteria and these leaf surface bacteria that have the most similarity to specific abundant sequences on the leaf surface RNA, with the idea that you would be getting base pairing between the leaf surface RNAs and messenger RNAs in the bacteria. Because if it's direct base pairing, then you have a chance to interfere with translation. And so that's our leading model right now for how we're inhibiting bacterial growth. But we're just now getting to those experiments.
Well, I would also like to interpret it a different way and then try to address it in the way maybe the question could also be that whether the leaf surface RNA is being contributed by the leaf isolated bacterial species. So to see the similarity at the level of, the contribution of RNA on the leaf surface by the bacteria. But going back to Jean's question about the notobiotic systems. So maybe that will allow us to say how much, is being contributed by the bacteria and how much is not.
Do you do you see bacterial RNA in your reads?
Um, Not a lot. But it's, it's a bit tricky because we don't have, a database of all the bacteria. And so certainly we can talk about what percent of the reads map to the host plant versus don't map. And you would guess that some of the ones that don't map are mapping to bacteria. The original experiment that we did, we purposely did not filter. And so with those RNA sequences, we see lots of bacterial RNAs, but those are probably coming from intact bacteria. So when
Yeah.
we filter and get rid of the intact bacteria, we see really relatively few bacterial reads.
Yeah.
So I don't think there's a lot relative to the plant RNA on the leaf surface. There's not a lot of bacterial free RNA on the leaf surface. Now
Cool.
that said, we have an ongoing experiment with a collaborator in India who visited my lab on a sabbatical visit, Anna Punagash, and she has found when you infect with Ustilagomatus corn leaves, you can actually find Ustilago RNA on the leaf surface, even though you filtered out any intact spores.
Mm.
So fungi certainly are secreting RNA onto the leaf surface.
Okay. Now we're going to talk about cross-kingdom communication and translocation, how RNAs and EVs interact physically with fungi and transport across plant species. So there are several consolidated questions regarding the cross-kingdom mechanics. Sungil Kim asked whether plant EVs are truly internalized by fungal cells or if they just remain attached to the fungal cell surface.
Okay, so here again, I'll define EVs as vesicles smaller than 200 nanometers because that's kind of what the classic work has been so far. And I think it's unlikely that an intact EVAC would be internalized just because they're so big. So years ago, Brian Rudder in my group did a lot of work with plant EVs looking for uptake by various fungal species. And he saw very strong adhesion to the surface of especially Colletotricum and Botrytis. But he had a hard time seeing any clear evidence of uptake so that it could cross the plasma membrane of these fungi. So based on his work, we were not convinced that there's really a high rate of uptake of intact vesicles. But on the other hand, we know these fungi take up naked RNA and does not require vesicles to take up the naked RNA. So our current thinking is, at least relative to our transkingdom RNA silencing, that's more likely to be mediated by RNA that's released from these EVACs than by uptake of EVs. Now, we certainly can't rule out that some EVs are taken up, but in our hands, we did not see compelling evidence for uptake of intact EVs. It's possible they fuse with the plasma membrane of the fungi, but looking for like bright puncta in the cytoplasm of the fungus, we just didn't see it.
So expanding on translocation, Mohammed Davaheri noted that in examples like Fusarium head blight in wheat, EVs have been shown not to be involved in double-stranded RNA translocation. Could you elaborate on other proven scenarios for double-stranded RNA translocation? And do you think EVAC involvement is highly plant dependent?
Yeah, so our data would definitely indicate that EVs do not transport much in the way of RNA into microbes. And so our current favorite model is that EVACs get secreted, get broken, and then release EVs onto the leaf surface. Those then, I'm sorry, release EVs, but also release RNA, most importantly. So they essentially are releasing RNA or RNA protein complexes onto the leaf surface. And then that probably does associate with polysaccharides, and it may be these condensates that get taken up directly by the fungus. And that, we need to do some additional testing, but based on our Higgs experiments, we know that the fungus is taking up RNA. And we know this RNA can be found outside of EVs.
Just remind us what Higgs is again.
Host-induced gene silencing. So that's when you express a double-stranded RNA in a host plant, and the plant secretes that double-stranded RNA. And that double-stranded RNA then is taken up by an insect or a fungus or oomycete, and silences homologous genes in the target organism. So go back to that question again really quick.
Right.
So proven scenarios. So we know spray-induced gene silencing works, meaning that you can just spray naked double-stranded RNA onto a leaf surface, and that RNA will be taken up by fungi and will silence the matching genes in the fungus. So we know fungi have the ability to take up RNA from the environment. And so to my mind, and the simple thing is plants just have to secrete RNA onto the leaf surface and fungi will take it up. You don't really need EVs for it to happen.
But that's the way it does happen, I guess. Yeah.
Yeah, no, I mean, I think fungi
Need is,
are
yeah.
good at taking up RNA from the environment.
Okay, so Zahar Mir, does the inhibitory nature of these RNAs on pathogens work through transcriptional inhibition or a completely different mechanism?
So it depends on the organism you're talking about. If we're talking about fungi, I think it's quite likely it engages with the classic RNA silencing machinery in fungi. So most fungi have dicer-like enzymes that chop up double-stranded RNAs, argonaute proteins in that bind short single-stranded RNAs that then would degrade the complementary messenger RNAs in the host. So that would be just classic RNA silencing, which then could inhibit growth. Bacteria do not have classic dicer-like proteins. Some of them have argonaute proteins, but not all. The best work on this has actually been done by Minu's PhD lab, and she contributed to it, where they were able to show that bacteria can take up short double-stranded RNAs from the environment, and that actually seems to inhibit at the level of translation rather than transcription. And so there, what is thought to be happening is these RNAs are base pairing because it was very sequence specific. So they're base pairing with the messenger RNAs, and that's somehow just inhibiting translation.
Okay, fantastic.
By the way, that's Leonel Navarro, if I didn't say that. It was his group that did that work.
Thank you. Back to Georgina.
Okay, let's go for the last set of consolidated questions. These ones are going to be about cellular mechanics, EV fusion, and cell wall penetration. Sung-Hil Kim noted that when isolating these vesicles at high concentrations, you don't seem to observe a fusion between them. Some part of this question was already answered in the webinar, but maybe if you want to elaborate.
Yeah, I'll just repeat the answer to that question. So we know these vesicles are negatively charged because of phospholipids on the cells on the surface of the vesicles, but also they appear to be coated with polysaccharides. And we know plasma membranes have a lot of highly glycosylated proteins on the surface of the plasma membrane, and we think the EVACs, most of them probably have a plasma membrane outer coat. So I think it's a combination of negative charge and these polysaccharides, which also carry charges that just keep them they kind of repel each other because they all have that same negative charge.
Great. Okay, there was another one that Sumya Gul asking about the isolation process and says, if ROR2 PEN1 is still detected in the purified EV fraction after filtration and centrifugation, does that imply filtration doesn't cause protein degradation, but RNA molecules are preferentially degraded in EVACs?
So certainly the proteins inside the EVs are quite stable through the isolation process, through the centrifugation and filtration steps. We actually think inside EVACs, the RNA molecules are quite stable. We've done some comparisons of these purified EVACs RNA that we get from purified EVACs and compare that to RNA that you would get in the apoplast, and it's much less degraded than the RNA in the apoplast. And it looks really pretty similar to whole cell lysate RNA. So we think the RNA inside the EVACs is pretty stable. It's only once the EVACs break open and RNA gets exposed to the environment that it starts getting degraded.
Right, perfect. Yeah, people can see the gels in your webinar. And then one more question about these large EVACs that you mentioned between 1 and 5 microns. If these EVACs can squeeze out from the plant cell wall and cross the cell wall matrix, is it possible that microbes, like fungi, generate EVACs and they send them through the cell wall to the plant? Or you have never seen, like, in
That's a really cool idea. We've not looked for that yet. I mean, we would need to generate a fungal line in which we have labeled the vacuolar membranes in the fungus with a GFP, so that we could really look for these EVACs. And that's just not something we've gotten to yet. So many experiments still to do.
Sure. Many ideas. And I don't
I like that
remember if we have-
idea.
But following up on that, Roger, is the vacuole of fungal pathogens or fungi in general are big like plants, or
They have large vacuoles, but they tend not to just be a single vacuole in a fungal
oh,
cell.
that's what we see as multiple bulbs.
Yeah.
Might happen. Well, nice to have the methods to look at that, because the electron microscopy big section. Okay, I think we are finished with all the consolidated questions we had for this webinar. So
Right.
Does anyone
Let's-
have any other thoughts about anything you want to amplify on, Roger? Maybe you could just give us a brief recap of your thoughts about this whole.
Well, I guess obviously we're very excited about this work overall because we feel like we've stumbled on a completely new pathway of secretion in plant cells. There's so-called unconventional protein secretion that's been known for decades, that a lot of proteins in the apoplast do not have classic signal peptides on them. So how do they get out? And so we think this is an explanation for how a lot of those proteins that you find in the apoplast that lack signal peptides are getting out, is through this process. And it's very unexpected. Like, how does the tonoplast membrane fuse with the plasma membrane to release these EVACs? And how do you end up with a plasma membrane potentially on the outside of these EVACs? It's like some sort of pinching out. And, from a cell biology standpoint, that's really puzzling, how you would push out these bags from the plasma membrane and pinch them off. And so I think there's a lot of really basic cell biology that is going to have to be addressed going forward here, that I think is going to be really interesting for the community to pursue, because nobody knew this even happened before. So
I think it's like a waste pathway, like, let's get rid of all this stuff. We're done with this, let's just ship it out. Or do you think it's
I think it's much more precise than that.
Yeah.
I mean, it's been known for quite a while that there are really defined attachment sites between ER membranes and plasma membranes, and even between tonoplast membrane and plasma membrane. But exactly what controls those attachment sites has been poorly understood in their purpose. And now I'm thinking that may just be for the tonoplast plasma membrane attachment sites, that just may be a seed location for where you can then secrete one of these vacuolar bulbs, which becomes the EVAC. And if our hypothesis is correct that the leaf surface RNA is really important for homeostasis of the microbiome, then that would argue that this is a really controlled process, that this is one way the plants have for getting that RNA out there. So, right now we're very focused on that hypothesis, but I think we're going to learn a lot of interesting new basic plant cell biology by studying that process.
great? So I'll just end here and thank again our sponsors. This webinar series is going to continue. Please, everybody who's interested, watch our social media. Also, check our website, which is going to be online very soon. And for the early career people, the advanced students and postdocs, we have a special series, a Rising Star series for you. And we're accepting applications right now. So you can use the QR code or contact us using this email. So thanks again to Roger and our board members for this addendum. I think it's been a fascinating discussion, and I think we may continue with this format. It's really nice.
Yeah, thank you, Jean. I really enjoyed this.
Yeah, great. Thanks.
Good.
Thanks
Bye,
all.
everybody.
Bye. Thank you.